In today's video, we're going to be taking a look at selection and variation for A-level biology. If you see a certain point that you want to skip to on this piece of paper, take a look in the video description below as I've listed all of the respective timestamps. Otherwise, let's go ahead and get started. Now, the first thing to understand is the relationship between genotype, phenotype, and environmental factors. So genotype refers to the genetic makeup of an organism, and the genotype provides a blueprint which provides the limits to which an organism may develop to. The phenotype is the physical makeup of an organism, and this will be produced as a result of a combination between the genotype and the environmental factors. Now, environmental factors are the conditions in which an organism lives in. So its environment and the outside conditions. For example, it could also be its diet, the temperature that it lives in. Now, let's take a look at the environmental impact on phenotype with these three following examples. So in Siamese cats and Himalayan rabbits, the recessive allele CS in Siamese cats and CH in Himalayan rabbits codes for a heat-sensitive form of the enzyme tyrosinase, making it unable to function at temperatures greater than 33 degrees Celsius. Now, tyrosinase is involved in the production of the dark pigment melanin. Now, on the body surface temperature of Siamese cats and Himalayan rabbits in general is greater than 33 degrees Celsius. So in these regions where the body surface temperature is greater than 33 degrees Celsius, the heat-sensitive form of the tyrosinase enzyme can't function, and so the fur color will be white. At extremities of these organisms, for example, the ears, the nose tips, the paws, where the body surface temperature is less than 33 degrees Celsius, the heat-sensitive form of the tyrosinase enzyme will be able to function, and so the dark pigment melanin will be able to be produced, and so the fur will appear darker colored. Now, the dominant allele produces the enzyme tyrosinase, which can function at a wider range of temperatures. So it's just this recessive allele that will produce that heat-sensitive form of the tyrosinase enzyme. Now, in Arctic foxes, they produce fur pigments which create a dark color, a dark fur color. However, in very cold temperatures, the dark fur pigments aren't produced, they can't be produced, and this leads in the fur color being white, right? Because if we don't have those fur pigments, the fur will just be white. And this is actually very, very beneficial because in those cold temperatures, so when the outside environment, the environmental factor is giving us a very cold temperature, the Arctic fox will remain white, and this is very beneficial because that means that it will be able to camouflage, right? If there is a snowy environment, if it's very cold and the Arctic fox has its fur being white, it will be able to camouflage and it will have a greater chance of survival. So that's an example, that's another example of the environmental factors on the animal's phenotype. In human height, if we think about it, our genotype, for example, could say that we should grow 1 meter and 90 centimeters. But if we aren't eating the correct food, if we aren't eating a nutritious diet, especially in the adolescent ages, we're not going to reach our maximum potential. So again, this is an environmental factor, right? The diet, the things that are outside of the organism. So now let's take a look at the difference between continuous and discontinuous variation. So characteristics that have continuous variation show intermediate levels between one another. They have a graded effect. For example, human height, right? We can be 170 centimeters tall, 170.5 centimeters tall, 171 centimeters tall, etc. On the other hand, discontinuous variation, the traits that have discontinuous variation have no intermediate groups. You're either one or the other. For example, blood type in humans. You can be A, B, O, or AB. Okay, there is no graded effect between the different groups. Now, polygenes occurs in characteristics that show continuous variation. So polygenes means that multiple genes are chipping in to the phenotype of that characteristic that shows continuous variation. Whereas characteristics that show discontinuous variation are normally coded for by single genes. So they're either one or the other. So let's take a look at some causes of variation. The first causes of variation are the genetic differences. So genetic differences can come from mutations in the DNA base sequence. We can have crossing over during prophase 1 of meiosis 1. We can have random orientation of homologous chromosomes in metaphase 1 of meiosis 1. We can have random mating, okay, so any two different individuals of a species can come together and mate. And then we have the random fusion of gametes during fertilization. Now, for these points that we discussed that are part of meiosis 1, take a look at the meiosis video as we go into more depth and how those actually result in variation. Now, on top of these genetic related sources of variation, there are also the environmental factors, right? As we discussed just before, the environmental factors like temperature and like diet, for example, will cause variation in an environment. If we have two organisms that are exactly identical, let's say hypothetically, they're both exactly identical, and to one we give them a very nutritious diet, and to the other we don't give them a nutritious diet, this first one will likely grow more than the second one that didn't have a nutritious diet. So again, that is how the environmental factors will be a cause of variation. So now let's take a look at natural selection. We have the three main points of natural selection. The first is that we will have overproduction of offspring. Now, within the species, within the population, there will be genetic variation. So the different organisms will have different traits, different alleles. Now, because of that overproduction, there will be a fight, right? There will be intraspecific competition for resources like food, like space to live, maybe shelter, maybe mates. Okay, so we have this overproduction of offspring, we have variation between all of the individuals, and we have competition. So what's going to happen is that individuals with favorable alleles will be able to win in the struggle to survive against the individuals that have less favorable alleles. Okay, so it's survival of the fittest. Now, what this is going to do is that over many generations, the individuals that have more favorable alleles will have a higher chance of passing on their alleles to the next generations than the individuals that have less favorable alleles. Okay, so what this is going to mean is that over many generations, the more favorable allele will increase in frequency compared to the less favorable alleles. Now let's take a look at the three types of selection. We have directional selection, where we have that intermediate phenotype, the average phenotype, and directional selection will be when either of the extreme phenotypes is favored over that intermediate phenotype. Stabilizing selection is when the intermediate phenotype is favored over the two extreme phenotypes. Okay, so we will just see that the extreme phenotypes will reduce in number, and that intermediate phenotype will be the most advantageous. Disruptive selection is when the intermediate phenotype is the least favored and the extreme phenotypes are more favored. They're better adapted to the given environment. Now, the disruptive selection is the type of selection that is most likely to bring about evolution, because with this we have the extreme phenotypes in action within the population, and this will increase the chance of evolution into a new species or a new subspecies. Now, gene pool refers to the total number of all of the different alleles of all of the different genes within a population at a specific point in time. And allele frequency refers to the frequency of each different allele within that gene pool. Now that we've taken a look at the gene pool and allele frequency, let's take a look at some processes that affect allele frequency. And the first example will be bacterial antibiotic resistance. So let's say we have two individuals. We have individual A and we have individual B. Both individuals are infected with a bacteria and they both get sick. Both of them receive an antibiotic treatment course. Individual A completes the treatment course and is healed. Individual B, on the other hand, after a few days, they feel better and they stop the treatment, leaving a reservoir of bacteria within them. What is then going to happen is that those bacteria will divide and they will increase in population. Now, between each division of those bacteria, there is a possibility of a chance mutation. And in a possible case, what may happen is that there may be a chance mutation that results in antibiotic resistance. So now we've got an antibiotic resistant strain of bacteria. The person, right, so once the bacterial reservoir has now grown in population, the person gets sick again and they once again get treated with the antibiotic. This time, only the bacteria that are not that don't have the antibiotic resistance will pass away, and the bacteria that have the antibiotic resistance by way of a chance mutation will survive. And now what we have is a very big problem, okay, because we have a resistant strain of bacteria, and this could be a global pandemic, right? It could be a global issue. Now, it's important to note that the second time that the individual was infected and we had the antibiotic resistant bacteria and we had the normal non-antibiotic resistant bacteria, those antibiotics that were put into the individual acted as a selection pressure, because it's an environmental factor that favors one individual over another, that favors the genes, the alleles of one individual over another, right? The antibiotic resistant bacteria were favored to survive over the non-antibiotic resistant bacteria. Now, genetic drift refers to the change in allele frequency over time due to random events. And it's a very subtle change. Genetic drift doesn't really occur. You can't really see the changes from one generation to the next. But two examples of genetic drift that are very powerful, that you can definitely see from one generation to the next, is firstly the bottleneck effect, where there is a significant reduction in the gene pool of a species, normally because a lot of individuals of the species will be killed, and so only a small population will be left, and that small population, of course, has a very small gene pool in comparison to the original full population. Something that could cause the bottleneck effect could, for example, be a wildfire, where most of the individuals of the species pass away, or right, and then those individuals will then continue the species forwards, but only with their genes, which is a very small gene pool in comparison to the initial full species gene pool. Another example could be hunting by humans, right? Humans could kill animals, could kill a species of animal so much that only a few individuals are left. And in comparison to the initial gene pool of the species, those individuals only have maybe a nail size worth of genetic variety, right, of a gene pool in comparison to the initial gene pool of the species. The founder effect occurs when individuals from one species will migrate to a new place and they will start up a new population in that area. Now, the new population will be based on the foundation of only a small gene pool, okay? So they're not going to have much genetic variety. The gene pool will be much smaller in comparison to that of the initial huge gene pool that the individuals left from. So now let's take a look at the Hardy-Weinberg principle, which is used to calculate the allele frequency of a certain gene from a population. Now, there are 5 criteria which must be met. The first one is that no mutations occur. The second is that the population is isolated so that there is no influx or outflow of alleles from the population. The third one is that there is no selection pressure, right? So that each allele has the same likelihood of being passed to the next generation as another allele. The fourth criteria, the fourth point, is that the population must be large so that there is a smaller risk of a chance event having an effect on the allele frequency. And the fifth and final point of our criteria is that mating within the population is random. Now, the formula, the Hardy-Weinberg principle formula is that p squared plus 2pq plus q squared equals 1.0, so is equal to 1, where p, for example, is going to be the dominant allele of where p is going to be the dominant allele and q will be the recessive allele. Okay, so that is going to be the Hardy-Weinberg principle. So now that we've taken a look at natural selection, let's take a look at artificial selection, which is also known as selective breeding. Now we have 2 types. We have outbreeding and we have inbreeding. Outbreeding is when we cross individuals that are not related to one another. And what this will do is it could result in offspring that are stronger and better adapted to survive than what their parents were. Okay, and this would mean that they have hybrid vigor. Okay, the hybrid vigor is created as a result of that outbreeding if the offspring have traits that make them better adapted to survive than what their parents were. Inbreeding, on the other hand, is when we cross individuals that are very closely related to one another, and we would normally do this if we want to maintain a certain trait between generations, right? We want to continue a specific trait through the future generations. The issue with inbreeding is that because we are crossing individuals that are closely related to one another, hybrid vigor is lost, okay? And this is because there will be a smaller gene pool and the fertility will be reduced of those interbred offspring. Now, this is actually called inbreeding depression, okay? So instead of having hybrid vigor, we're going to have inbreeding depression. Progeny testing is when we test, right? So when we store the characteristics and traits of offspring of individuals. Okay, so if, for example, we wanted to do progeny testing for a specific bull, we would see what sort of offspring has it created in the past. Has it created offspring that are that produce a lot of milk? Has it produced offspring that are very large in size, right? And so we store these traits, and then we can use that information to make decisions on possible selective breeding in the future using that specific individual. So that's progeny testing. Now, selective breeding for increased milk production in cows can be done, right? So using our progeny testing, we can identify which individuals have produced offspring that produce favorable features like high yield of milk. And with that in mind, right, so once we have the 2 individuals that we want to cross, a cow and a bull, what we can then do is we can extract the semen from the bull. We can store it if we need to, right, cryopreservation, and then we can fertilize the oocytes, the eggs of the cow via artificial insemination. Okay, so that is how we could do selective breeding for an increase in milk production in cows. So some rice is affected by the fungal disease called blast, and this can reduce the crop yield by up to 50%. So what breeders can do in order to find disease-resistant rice to blast is they can breed, so they can screen, and then they can breed rice to try and find a blast-resistant variant of rice, which is what they've done, and they can produce that in a larger scale, right? So to produce disease resistance in rice in order to increase the crop yield. Now, interbreed, so here it's actually supposed to be inbreeding, okay? So we have inbreeding and hybridization in maize. So what happened is over a long period of time, farmers naturally started, they wanted one specific trait of maize, so they inbred it, okay, because they wanted that favorable trait. Now, what happened is that over many generations, the inbred maize, so what happened was the inbreeding depression. The inbred types of maize decreased in productivity, so they started to not be as efficient as before. Now, what people started to do is that they started to combine inbred types, right? So they started to do hybridization. So hybridization is when we combine two different organisms with one another. And what that led to was increase in crop yield, okay? And so that is how inbreeding and hybridization actually enabled the maize production, the yield of maize to continue being high